pritisnil. Zdaj ne vem, snema, zgleda, da snima.
Ok, super. Hello everybody. It's my pleasure to introduce our guest, Marinka Žitnik. So thank you for accepting our invitation. She obtained her PhD in computer science at the University of Ljubljana in 2015. And in 2016 she started working as a postdoc researcher at Stanford. Hello. I probably should have mentioned also your rewards, but we are running late and the audience is here for you, not for me, so I will give you the word.
Okay, thank you, Nina. Thank you for this nice introduction. I'm very happy to be here and to be able to give a talk and meet you all. So I'm happy to take questions during the talk or after the talk, whatever works for you. And I'm happy to take questions in Slovenian or English. Today I will talk about deep learning for network medicine. In particular, it will be a computationally oriented talk, but we will be I'll be describing methods that tackle some of the biomedically important problems. So let me start with a very high level motivation. You are probably some of you here are fans of science fiction, and when somebody mentions AI and medicine together, you might think of some of the very futuristic use and devices and opportunities that AI has for medicine, for example, in the sense of having some devices that are able to measure the human body and various properties and statuses of molecules, as well as other aspects of health, and then doing diagnostics based on that, or having some kind of robotic assistance during procedures for surgery or something like that. So of course this is science fiction, but there has been a tremendous progress in the last decade or so in the sense of new technologies that now allow us to improve the medicine and discover either new science to advance our understanding of nature, or to really improve the healthcare in terms of diagnostics and prognostics for people. And so here I'm just listing, it's not of course comprehensive list, but it's a list of different devices that generate data that describe various aspects of human health and body. This can be various kinds of imaging data, it can be patient sample profiling, DNA sequencing, DNA microarrays are really one technology that has generated tremendous amounts of data in the last decade. Then various molecular diagnostics, measurements, wearables that come in the sense of smart watches or smart lenses and so on. Advanced prosthetics, of course, electronic medical records are now really a very popular and source of large amounts of data that need to be understand better. Prescription and prescription data, as well as various molecular technologies such as CRISPR and others that have allowed us to engineer genomes and things like that. So medicine is really revolutionized by these new technologies, and what these technologies have allowed us, they have enabled us to get new insights into human health and disease and our bodies. So there are lots of opportunities and there are lots of benefits of having these technologies. We can really push forward science in the sense of we can understand now better how drugs work, how to treat the diseases, and how does certain human processes such as aging or health change across populations, as well as these technologies have led to improved care and improved research and development, for example, in drug discovery. On the other hand, these technologies really generate massive amounts of data. There is this estimate that medical data is expected to double in size every 73 days by 2020. So this really means that now the amount of data is so huge that it's impossible to just look at the data and see the patterns, or really to even analyze the data without really deeply thinking about what kind of analysis can we do and what will this data analysis reveal to us. So there is a lack of computational models, there is a lack of large-scale analysis of this data. In a way, I often think about this is in the sense that science has really been, in general, enabled by really development of new scientific instruments. So in the past, there has been microscope, and just the design and invention of microscope has really led to lots of new advancements on various aspects of science. And what we are now is in some very similar time where we need new instruments that will allow us to really make new progress for modern data-intensive disciplines, and medicine is becoming one of those. So what are the main challenges here? So the data is being generated at this unprecedented scale, and it often happens that for a particular new experiment or a new research question, domain experts generate a data set. That data set is used to answer that research question, and then different group or different lab moves on and generate a new data set. So it's really, these data sets are being generated. That's really an expensive and time-consuming process, but often the data are not necessarily being leveraged to inform further downstream future experiments. Also, the data here involves rich multimodal relationships and interactions. Often it is the case that we have, for a particular problem that we care about, such as disease, there are many different types of data set that might be relevant for that problem, and this data might be recorded at different levels, so it's not really easy to think about how to integrate that representation and come up with a model that will allow us to do some predictive modeling. The high-level approach of my research is to leverage these data sets at the scale of billions of interactions between millions of various biomedical entities, such as patients, drugs, and diseases, and I will give an example of that in a few minutes. And then develop learning methods that operate on these data sets in order to generate actionable insights. So this was a high-level introduction, and now in the rest of this talk I will talk about one specific recent project that I worked on, and that was the project for developing deep learning methods for modeling drug combinations. Okay, so what is motivation for this work? So of course, when we have a certain disease, we go to doctor, and we get a prescription, and we take drugs typically. And the way these drugs work at the high level, we can think of them so that the drug is being administered to a human, and then the drug affects the activity of the body by actually binding to certain proteins, certain molecules in human cells, and then it alters activity of those proteins. As a result of that, hopefully disease will be cured, or we will achieve some desired therapeutic effect. Right? So this is the positive outcome. Unfortunately, when a patient takes a drug, something bad can happen as well. And these bad aspects are unwanted side effects. So let me give a small example. So imagine we have a large reporting system that actually records different types of these unwanted side effects for different types of drugs that people take. So here in this system, let's say we have now 3 different drugs: this red one, green one, and orange drug. And what this list is about, it says when patients take red drug, then these are 3 types of side effects that they experience, and 5% would be the side effect that would be the percentage of patients that experience this type of side effect. And 10% of patients on the red drug have experienced increased heart rate, and 3% of them really suffer from some kind of aneurysm. So there is a different drug, of course, that exists on the market that is taken by different or the same patients, and that can lead to some other types of side effects. So the nice thing, or kind of these are side effects of individual drugs. Here in the last 2 rows, we know that, well, if patients will take orange and green drug together, then they will have more side effects. And these will be the side effects that they can experience. Or if they take red or green drug together, then there are some other side effects. So what is interesting in this list? There are a few interesting things here that I want to highlight. So first is, well, if we know what are side effects of a red drug and or a green drug, does that mean that we know what will be the side effects that a person will experience if a person takes both drugs together? So intuitively you could think of, yes, we know, because we would just assume that, well, if you take these two drugs together, the side effects that you will experience are just the union of side effects that you would get if you take either drug alone, right? And for this pair, this actually is the case, right? So these are the three side effects from the red drug and the three side effects from the orange drug. What is interesting, however, in this particular other case, this particular other combination of these two drugs, there are some new additional side effects that patients experience when those side effects are taken. So these are additional side effects that patients do not have if they take just orange drug alone or just green drug alone, right? So why do we have these side effects here? Sort of, this problem is known in the literature as the problem of drug interactions. So this is the problem where we observe certain effects in human body, and this might be desired or unwanted effects, and those effects are observed only when a particular combination of drugs is administered. And this is unexpected because one would not expect that if there would be no kind of interaction between drugs, and drugs would be independent of each other. So just to reiterate again, so the idea is here, in the simplest case, when the patient takes either drug, no side effects. When both drugs are taken together, some side effects. And that's the problem of drug-drug interactions. So is this really an important problem to tackle or to understand, or is this really something that happens very rarely? So it turns out that this is really an important problem for which the current medical experiments or the clinical trials really cannot model and analyze it well. So the problem is known as polypharmacy, which refers just to the fact that the patients take more than one drug, multiple drugs at the same time. This is common to treat complex coexisting diseases. So how common it is? It's actually around half of population over 65 years take more than 5 drugs at the same time. And many take 20 drugs to treat very complex diseases such as heart disease, depression, cancer. And at least in the US, these kind of side effects affect around 15% of the population, and there's a significant healthcare cost associated with treating unwanted side effects. So this seems to be an important problem. So then the question is, well, if this is an important problem, can we actually detect these unwanted side effects of drug combinations before drug combinations are actually just prescribed in real patients, and then we can only observe what are all bad things that happen to patients. There are some challenges that we need to really address if we want to do that. So what are some of the challenges? So of course you can assume that there are many different types of side effects, right? So there is nausea, different types of pain, or there might be some joint stiffness, and like all other the side effects that were also mentioned in that example from that I showed a few slides ago. So each side effect occurs only in a small subset of all patients. So very quickly what it happens, we have a very large data set, but if we focus on any specific side effect, then only a small population of patients are really relevant for that side effect, right? Then, of course, side effects are not independent of each other. One could assume that side effects that affect cardiovascular system might have some common biological underpinning. So can we actually model that? The problem is that with combination of drugs, we quickly have the problem of simply the explosion of possible combinations of drugs. So currently on the US market, there are around 5,000 approved drugs. If you just look at pairs of drugs, that's 5,000 choose 2, right, pairs of drugs. So that would mean when a pharma company would like to get approved from FDA for a new drug, they would need to run 5,000 experiments to compare that new drug with all drugs that are already on the market. And that's impossible to run now 5,000 clinical trials for that. And even if they would do that, they only tested for pairs of drugs, right? So for like 2 drugs. But what about like three drugs together and so on? So it's a problem that really cannot be just tested by using experiments. Of course, there are different types of data that might be relevant for this problem, like adverse events and side effects that patients are reporting already for drugs that are already on the US market, as well as various kinds of data that describes how actually drugs work. So how can we take this data together in order to model the problem? So what do I mean by model the problem? What we'll do today is actually I'll describe a system that the simplest case you can think of is as a system that allow us to answer a specific type of queries. And this type of queries that we'll be answering is given a particular combination of drugs, let's say drugs C and D, and a particular side effect R, we'll answer, well, how likely it is that combination of drug or pairs of drugs C, D leads to side effect R. So this will be the kind of queries that the system will answer. Given a combination of drug, given a side effect, how likely was the fraction of patients that will have that side effect? Okay, so the problem formulation that we will use here is really we will leverage the power of graphs as a very important flexible language to represent our data set. And in particular, in our case, we will denote the knowledge of side effects of a particular drug combination and we represent that as labeled edges in our graph. So in this particular example, we have 2 nodes here, C and D. These 2 nodes represent 2 drugs that are on the market. And if we know that these 2 drugs, C and D, lead to R2 side effect, which is bradycardia side effect in our model, then we will add an edge here. So this means that when a patient takes C and D together, R2 is likely to occur. And R2 would not be a side effect that someone would get if the person would take only C or only D. Okay, so of course we don't have just 2 systems, two drugs, right? But there are many other drugs, and C might also interact together with other drug S. And if C and S are taken together, there might be a set of different side effects. And with C and M are taken together, there might be some other subset of side effects that one would experience. Also, this is not the only type of data that we have, but really what is important also is, well, what really happens when a particular drug is taken by a patient. If you remember before, I said that when a patient takes a drug, drugs bind to certain molecules in human body, and then those molecules change their activity, hopefully in the way that it will be beneficial for patient. So to encode this knowledge, we represent information on what are the kind of molecules or what are the proteins that are targeted by a specific drug. So here we know the drug C targets 4 orange circles. These are 4 different human proteins in the human body, and then these proteins might interact with each other in some way, and these are these edges between orange circles. Of course, that's not for C, but for all different drugs, and these are not just these 4 proteins, but all proteins that human has, and by doing that, we get this large multimodal graph that consists of drugs and their relationships and side effects that they might lead to, and molecular level information on the interactions between proteins. So in practice, to really instantiate and construct this graph, what we use is large patient molecular and drug data. So in particular, we take the human protein-protein interaction network that indicates physical interactions between proteins. We take these drug target relationships so that we know for a particular drug that is approved on the market what are the proteins that the drug is targeting. Then for side effects of drug pairs, we focus this, we take the National Adverse Event Reporting System. That's a reporting system in the US that is used by doctors, so that when a patient comes to a doctor and reports a particular type of side effect, then the doctor will record that information in that national adverse event reporting system. And then that information is aggregated, and that allows us to construct the drug-drug interaction edges. And then we have various side or auxiliary information on drugs and proteins, and the way you can think of that is we have some feature vectors on proteins and drugs, so that are additional information on the behavior of or properties of drugs, such as the chemical structure of the drug and so on. So altogether, this results in this multimodal network that has around 5 million edges and 1,000 different types of edges. So it's highly multi-relational in the sense that there are many different types of edges, and those edges are different types of side effects. So if we have this formulation, then what is our goal, or how we can kind of phrase our prediction test? We can say, well, given this partially observed graph that I have just described, the goal is to predict labeled edges between drug nodes, right? So in our example query, given drug C and D, we want to predict how likely it is that an edge between C and D of type R2 exists in our system. And that would mean that if C and D are co-prescribed, then this will lead to side effect R2. Okay, so what is the project we're using here? So the project that we have developed is an instance of graph neural network approach. So this is a class of neural networks that are designed for graph structure data. So you're probably familiar with neural networks that are designed for images and for text data, so convolutional neural networks and recurrent neural networks. And graph neural networks is another class of models that allow us to develop very accurate deep learning models that can operate on graphs directly. So at the high level, the way you can think about it is we take as input our multimodal graph, and then we will have a series of graph convolutions that will allow us to take that graph and compress it and learn some latent representations for nodes in that graph, and then leveraging those representations to finally make predictions and answer queries. So I will unpack this a bit in the next few slides. So there are two key steps of this approach, and perhaps the easiest way to think about it is in the following way. So we think of the first step as the encoding step. So that's the step where we'll take that graph and we will learn an embedding for every node in that graph. So what's an embedding? An embedding is, the way you can think of it, is a d-dimensional vector that captures the topological structure of a node. So it's a d-dimensional vector that can really represent local topology and interaction patterns of that node in the network. And we will do that for every node in the network. So this is the encoding step. So the idea is we take the graph and we encode it and embed it in some d-dimensional space. Then once we have that, the embeddings, then the second step is the decoding step. So in the decoding step, we will take this embedding space and all the embeddings that we have then, and we will learn how to combine them in a way to make predictions. So the simplest way to combine them here is just to take embeddings of two nodes, and then do simply a dot product or some bilinear form that will indicate the likelihood of an edge existing between the respective drug nodes. But there are many other types of decoders. So let me focus now first on the first part, on the encoder part. So what I mean here by embedding nodes a bit more. So by embedding nodes, what I mean here is we take as input a graph, we want to learn embeddings, as I said before. And what this means is that we need to learn some data transformation or mapping function, which will turn out to be a deep model of graph convolutions that will be able to capture some notion of network proximity. So what does that mean? We want to map nodes in that d-dimensional space. In this particular example, they're just two-dimensional space, such that if two nodes are proximal in the network, so are close together in the network, they will be embedded close together in that space. So the main question now becomes like, what does it mean when we say two nodes are similar or are close together in the network, that they should have similar embeddings? So this really depends on this notion of similarity between nodes that will really define the structure of this embedding space, depends on a particular application that you might have. In the simplest case, you might want to think that nodes that tend to have similar local network neighborhoods or similar network neighbors, then they should be embedded close together. So in this particular case, what I'm showing here on the right is an embedding space where every point is a different side effect. And then we see that there is some interesting non-trivial clustering structure of these points. And really what we can see by examining what are the side effects that these points represent is that we get these clusters of side effects that tend to co-occur in patients. So here's like one blue cluster, here's one orange cluster, one green cluster, red clustering, and these tend to be side effects that patients typically have when they take drug combinations. Okay, so really the question now is, well, how do we learn this function f that will embed similar nodes close together in that d-dimensional space? And that is achieved through this encoder process, and through a series of operators that are called graph convolutional operators. And the key idea is we will generate node embeddings based on the structure of their local network neighborhood. So imagine this is our current central node, which is one drug node, this green circle, and here I'm highlighting its direct neighbors and then two hop neighbors. And what we will do is, well, the idea is that we will learn how. So first we will think that every node in this local network neighborhood has some kind of vector message. And those vector messages are then being propagated and transformed along edges of those local network neighborhoods. So this means that what we'll do is every node here will learn how to receive those vector S messages from its neighbors and how to transform and how to aggregate them. The problem here is that these edges are not are heterogeneous in the sense of that they are different types of edges. So in order for us not to ignore different types of edges, what we will do is we will have a separate processing channel for every edge type. So what does that mean? So here I'm just showing this local network neighborhood for this green node, but only in edge type R2, which means that this green node has 4 neighbors. Out of 4 neighbors, two neighbors are defined by R2 edges, which means that these 2 neighbors will send their vector messages to this central node, and that node will learn how to transform, how to aggregate and transform those vector messages. And that will be the output of that central node. Here I have only taken its direct neighbors, but those other nodes will receive their own vector messages from their own direct neighbors. And the same is done for a different type of edges. So for R3, this central node has these 2 different neighbors of type R3. So it will receive vector messages from these 2 nodes. And those other nodes will receive their own vector messages from their neighbors, which are now 2 hop away from the central node. And then this will be non-linearly transformed. Okay, so the way you can think of it now is like, how do we actually get a deep model here? So the way we get a deep model is then graph convolution specifies for one layer how a particular node in the network will aggregate information from its direct neighbors, and then how that information will be transformed. That is used as output of the current layer and is used as input to the second layer. In the second layer, the node will be able to actually reach not only its direct neighbors, but neighbors that are 2 hops away. And by stacking together k-graph convolutions, this means we actually get a model where an embedding or this vector message for every node will depend on the structure of its k-hop neighborhood. Okay, so why are these graph convolutions actually, why is this a hard problem? Why can't we use just like a simple convolutional neural network or recurrent neural network that kind of everyone is using? Or that these are kind of models that are developed in modern like deep learning toolboxes like PyTorch or TensorFlow. It's really that these deep modern toolboxes are designed for grid data or for simple sequences, and graphs do not fit into that paradigm. So what I mean by grid data? By grid data, I mean images. You can think of images as being a simple graph, which is just a grid graph, where nodes are pixels, and every pixel has 4 neighbors if you have a single channel, or there are multiple neighbors if there are multiple RGB channels. But these are grid graphs. You can think of sequences like text or DNA sequences as having one linear 1D structure, and then if you have this linear 1D structure, it's very easy to define local network neighborhood, because these are defined simply by having a sliding window that is moved left or right? And the problem is that graphs are far more complex. We cannot just simply have this sliding window that will go 1 point or like 1 step to the left or 1 step to the right, because there's no notion of 1 step to the left or 1 step to the right. That's why we need these functions that can transform these local network neighborhoods, as I said. So what graph convolutions allow us is really to generalize convolutions that are traditionally defined on images or on sequences to more interesting structures and to more interesting graphs. So just to recap on the high level, graph neural networks are a type of neural networks designed for graph structure data that can take graphs as input. Through a series of graph convolutional operators, we can transform transform our network into and embed it into some low-dimensional space, and then use this low-dimensional embedding space to decode edges and make predictions. And really, what I'm not including here is the link, but it was there on the first slide. It's this class of neural networks now becoming very popular. There's lots of papers on this kind of class of models that's being proposed at recent major ML conferences. We had a few tutorials about these graph neural networks at DAP and ISMB this year, and all the materials are available online on the. You can just go to my website and you will find the links. So if you're interested into learning more about this, into trying out some models on your own data, you can do that, and I'd be happy to take questions about that. But now, let's assume we have this model. So when I now described the model briefly, and I described the data set and the graph that we have. So now it's the time to say, well, we have the model, and we have kind of the algorithm, and we have the input data. We want to train the model and then use it for prediction. So, of course, as any machine learning model, this graph neural network has many parameters. Those parameters need to be learned, and that's achieved through a process that is a training process of parameters based on our partially observed graph that we have. And in the testing phase, we use our trained model to answer queries about new drug combinations, such as the one that are in this form. Okay, so when we trained our model and we evaluated its performance against various updated versions or simplified versions of the model, our conclusions were the following. So that approach, which is called Decagon, was able to predict side effects for drugs with accuracy in terms of AUC around 0.8. That accuracy very much depended on different types of side effects. So the model was more able to predict side effects that are associated with various molecular events than side effects for which there might appear because just of behavior or lifestyle of a patient. That's not surprising because behavior and lifestyle of a patient was not part of our graph that I described. So basically the model did not have that information. What was interesting that model was able to work even for drug combinations that were not yet used in patients. So that's especially exciting because it gives us the opportunity to make certain predictions and flag unsafe combinations of drugs even before they are used in patients. So beyond just doing cross-validation studies, we wanted to ask two questions. Well, can we now use this method really to generate some hypotheses that will give doctors some guidance on whether it's a good idea to prescribe a certain combination of drugs to certain patients, or researchers some guidance on designing wet lab experiments. So we did that, and the way this was approached is in the simplest way. We first ask, well, what are new predictions that our model can make? Sort of predictions that are not in our data set, that are not yet known, and are considered to be highly, and the model is very confident about it. So here I'm showing top 10 predictions, and these predictions are the form that we have these pairs of drugs and different side effects that these pairs of drugs are associated with according to the model. So then we ask, well, can we first validate some of their predictions? And the first thing to do is to check, well, are there some reports of patients that are on these drugs and have side effects? Have there been any reports in the last year or so? So when we looked at these top 10 predictions, we found very recently there were several case reports reporting patients at various hospitals that were on these pairs of drugs and that have experienced side effects. And then we asked, well, what about these empty spots, right? Of course, those case reports are reports of a small number of patients, but can we actually, can we kind of do something more and validate our model in the clinic, not just retrospectively by looking at recent case reports that were published on PubMed. So the way to do that is then we ask this question and we ask, well, are predictions generated by the model, can they be actionable? Can some domain expert actually take our predictions and do something with them? So we teamed up with Mass General and Stanford Medicine School, and we asked them, can we use your medical records information in order to validate our predictions? So, and here I'll give a key idea of what I mean by validating that predictions in clinic. So assume that like a model makes the following prediction. The model says, well, the prediction is that the red and this orange drug will cause nausea, a side effect, right? This is prediction of the model. It's not yet known. Does it make sense? So the way this could be validated is by doing something that is known propensity score matching in the literature. But in the simplest case, it works the following. So we have a large database of patients, and then we look for triplets of patients that have very similar history. Let's say we have one triplet of patients, patient 1, patient 2, patient 3. They are very similar in their patient history, meaning the kind of diseases that they had in the past, perhaps their age, their occupation or lifestyle and behavior. And for these 3 patients, we know, well, patient 1 is similar to patient 2 and 3, and patient 1 has only taken this green drug, and afterward, and that's it, right? No orange drug. Patient 2 has only taken orange drug, no green drug. And patient 3 was actually a patient that was prescribed green and orange drug at the same time. And then we ask, well, say, after a while, only patient 3 was put on an anti-nausea med by a doctor. So why was patient 3 put on an anti-nausea med? Well, obviously he had some problem with nausea, right? That's why a doctor has prescribed anti-nausea medicine. But that was not the case for patient 1 and patient 2. So now, another explanation is that, well, patient 3 was put on an anti-nausea med because there was some other disease developed, some secondary disease developed in that patient, and because of that, a symptom of that disease was nausea. It could also be that the nausea was actually a side effect of this particular combination of drugs. So this is called propensity score matching because what we look for is almost we try to do causal inference on observational data. And I can talk more about this, but the idea is that we now look for many triplets of patients like that. And if we are able to find many triplets of patients like that, this gives some evidence that this prediction is fine, and kind of is quite high quality and kind of okay. And that's currently what we are doing to validate some of the predictions that the model has made. Okay, so I'll conclude in this 2 minutes or so. So in particular, I will summarize. What I have described today to you is an approach that was designed for the problem of drug combinations and predicting side effects of drug combinations, which is an important problem from the point of view that cannot be really tackled by just running more and more biomedical experiments. So it's really a problem where we need computational models, or we need machine learning models that can help guide domain experts. The approach that I described for doing that is a graph neural network approach. This is a general class of models that now allow us to apply neural networks much more broadly than just to images or sequence data. Since graph neural networks were introduced, we use them for many other applications. For example, to predict what are drugs that will treat a particular disease, to do various other molecular bioinformatics problems. There's a tutorial with lots of slides in kind of mathematical derivations of the model that you can check, in which is online. Okay, so finally, to summarize here, I want to say that, well, the way I think about these problems is that the science is inextricably connected to the existence of various scientific instruments. I think in the past we had these physical instruments, which are great, and we still have them, but now we also need a new class of instruments, which will be those data science machine learning tools that will become like a standard toolbox in some of the sciences. This will conclude my formal part of the talk, and I have one add. So there is a fellowship program. So there is a foundation that is called ASEF, which is American Slovenian Education Foundation. That foundation has fellowship programs on this website that allow students to visit universities in the US and also in England, Great Britain, and this year for the first time in New Zealand and Australia, and go into those universities for a summer visit or for around 10 weeks and work intensively on research projects that can then lead to publications and so on. So there are currently 32 universities that are in that program. Here's just a subset of those, most in the US. And I just want to say that the fellowship program is now open. There's a new call that is on the website. So if you go here, you can apply or learn more about what kind of projects you can do. Okay, and that's the end of my talk.
Okay, I'm sure there are plenty of questions. Who wants to start?
Okay, I have one. So you were talking about two drug combinations, right, and side effects. Have you tried your model on 3 or more combinations of drugs, and what are the results of that?
Yes, that's a great question. So first is how to even represent 3 drugs, because we represent them as edges. Yeah. So in that case, we represent them as hyper edges. So we train the model on hyper edges and make predictions for triplets of drugs. The problem is that it's basically impossible to validate predictions, at least in the standard machine learning way, where we would want to do some cross-validation or something like that, because the data on side effects of 3 drugs is so sparse that it's highly unreliable. So we have some case studies more or less for this, but no systematic evaluation of the method.
Okay. And the evaluation that you were talking that you're doing at Stanford Medicine, is this done by hand? I mean, how do you find all these records? Do they have a very good medical information system which can allow you that, or is that like?
So there is the entire department of biomedical informatics where there are researchers who work a lot on natural language processing for medical record data, so that the part of the data that does not necessarily come in the structured format, this kind of information on different types of drugs that patients are on, different types of side effects that patients are reported, can be extracted from text. The good part is also that at least for the last 20 years, these schools, they have quite good medical record system, so that drugs and times intervals and side effects are recorded using established international terminology, which is good because then we don't get additional errors in our evaluation just because of the problems with NLP. So it's not manual, but it's still non-trivial in the sense that it requires this propensity score matching to be actually deployed in the medical record system.
Okay. And how many cases do you want to have with the same side effects with drug combinations to say, okay, now this prediction that I got from the computer is great? Okay.
Yes. So we currently have a case project with prostate cancer patients. For that we have 60,000 patients, like for some of the most reliable predictions that were validated, which is quite good.
Yeah,
it's a lot. That's a lot of patients. For certain other diseases, we have at most hundreds of patients. So the reason why we have 16,000 patients for prostate cancer is because lots of very hard prostate cancer patients come to that hospital. So there's a huge hub of prostate cancer patients there. Yes.
Also got a question. So you talked about the combinations of drug effects that weren't predicted, that weren't observed before. So my question is, how many of those kind of combinations did you find, and how many were actually then later discovered as dangerous?
Yes. So that's exactly what we are trying to do by validating our predictions in the clinic. We are trying to validate our predictions by actually comparing predictions to experience of real life patients. And the other aspect is or like the simple computational study that computational experiment that we did was retrospective study. So basically what we did is we took National Adverse Event Reporting System in the US, which records all different types of side effects that patients are reporting. We trained our model on all the data up to 2010. And then we let the model make predictions, and we evaluated those predictions against the data that was reported and side effects reported between 2010 and 2008, right? And that were the numbers that I showed in that slide, where the AUC was around 0.8. These are the results for that kind of experiment.
So I think Glenn was asking as well, how many of those combinations that you found were not yet reported? Like, is that a number 10,000 or 1 million, or?
Because my guess is doctors know that this and this drug cannot mix together.
Yes.
Okay, but how many of those combinations they didn't know about, and you say now, this and this cannot be used together either? So
currently we have around hundreds. We have a few hundreds of this kind of drug combinations.
Few hundred?
Yes. That were combinations of drugs that make sense. So that, like, in principle, if you would be a doctor, you would actually you could imagine encountering a patient where somebody would actually want to prescribe these two drugs together, right? So because certain drug combinations do not make sense, right? No, but you would never prescribe them. But we have a few hundreds of examples where these are plausible drug combinations. The model has made some predictions that turned out to be predictions that were validated through these medical records or validated through case reports, but were not flagged on FDA, so the US Agency for Drug Administration, as unsafe drug combinations. So one direction of this research is basically also. We have started communicating with FDA on taking some of those predictions and potentially almost creating a prioritized list of what we think are unsafe drug combinations, so that FDA can focus more on those drug combinations. Because the drug combination space is so huge, then at least can we identify the part of the space of the drug combination space that is that's more unsafe, and that regulatory agencies can focus on that.
So here's an easy question.
Yes.
You showed us a nice slide about there was the red pill and the side effects, and there was some other pill.
Yes.
And then the combined side effects were different. Is this based on some real drug, or is it just to illustrate?
So yeah, that example that I showed you there was a hypothetical example,
Right.
but it was there to illustrate what is this problem of drug-drug interactions. That there are some new side effects that emerge when the combination drug is taken, because we have this non-additive effect of drugs.
This 4% colon cancer seems quite serious.
Yes, yes, it's serious. But that was just a made-up example for illustration.
Yes, please.
I have a few questions. The first one, but I'm not sure that I missed something. That when you have the transformation to D dimension, then what is the D? How do you know the D?
Yes, great question. I didn't go into that. So D is the dimensionality of latent space. It's a parameter that needs to be selected through. It's a hyperparameter of the model, so it needs to be selected through grid search or some other approach. This tends to be a parameter that deep models are not sensitive to. So if it's sufficiently large, what does that mean? Somewhere between 64 to 500, then the performance of the model will be good, and moving beyond that does not really lead to improvement in performance. But it's hyperparameter of a machine learning model. It needs to be selected in advance.
The second question is probably I know the answer, but so it is not possible to go forward with personalized medical stuff. So like concerning the condition of the different, so because different people can have a different side effect, different kind of medical conditions.
Yes.
Maybe it's not the same like when you would like to consider three effects, something is similar, yes?
Yes, that's a great point. That's absolutely true. The patient history really is important for predicting certain types of side effects. And because in the graph that I showed you, there's no notion of patient history, right? Because this is the fraction of patients that are on this drug combination and have experienced some side effect. By doing that, what we notice is that the model performs really well at predicting molecular side effects, like different types of molecular events, or that can be described actually by that happen because the molecules do not necessarily operate in the human body the way they should. But this model cannot predict that well side effects that are more explained, or where there are huge components of behavior or lifestyle of patients, just because where geographically that patient lives, or what's the kind of lifestyle that the patient leads. This is really important for whether a patient will see a certain type of side effect. So we are also now trying to incorporate the patient history information when predicting predictions, so that when doing predictions, so that predictions will be more personalized.
I have a third question.
Sure.
Just this deep learning. Okay, and is there any other methodology by which it could be compared the result?
Yes, so I didn't go into that, but we compared our method to some of the more traditional methods for multi-relational graphs. So these more traditional methods in our case were matrix factorizations and spectral methods, in that particular case tensor factorization, because you can think of each type of side effect being one adjacency matrix between drugs, right? One binary adjacency matrix. And then if we have 1,000 different types of side effects, we can get this huge tensor of 1,000 different matrices, right? And there are some models, like one prominent model is called Rayscale model by Max Nickel from MIT now at Facebook, where essentially you're running a tensor decomposition of that matrix. And there are also some standard work such as DDCOM or Parafac, the methods, where you would want to factorize that tensor. The problem is with those methods is, first the issue is scalability. When we have so many different types of side effects, and so many drugs or different drug therapies that we want to consider, it becomes very hard to factorize, decompose those large matrices. The second problem is that it's this notion that necessarily doing factorization of matrices is not the best approach for modeling graphs, because you want to capture this local topology, indirect neighbors, and kind of community structure for which these kind of models that operate directly on graph structure are really useful. So yeah, I'd be happy to talk more about this as well.
Okay, thank you. Is there any other question? If there are no questions, I suggest that we thank our speaker.